Rest now and let these gentle facts about the human brain accompany you into peaceful sleep. Your brain, that remarkable 3-pound universe nestled within your skull, continues its quiet work even as you drift into dreams. The human brain contains approximately 86 billion neurons, each one a tiny messenger carrying electrical signals across vast networks of connection. These neurons communicate through synapses, and your brain holds roughly 100 trillion of these connection points, creating a web of communication more complex than any computer system ever designed. As you relax deeper, consider that each neuron can form thousands of connections with other neurons, creating pathways for thought, memory, sensation, and consciousness itself. The brain consumes about 20% of your body's total energy, even though it represents only 2% of your body weight. This hungry organ requires a constant supply of oxygen and glucose to maintain its ceaseless activity. Even now, as you rest, your brain processes sensory information, consolidates memories, and performs essential maintenance tasks. The cerebral cortex, the wrinkled outer layer of your brain, houses the neural circuitry responsible for higher-order thinking, language, and conscious awareness. This thin sheet of tissue, only about 2 to 4 millimeters thick, contains the machinery that makes you uniquely human. Blood flows through your brain at a rate of approximately 750 milliliters per minute, delivering vital nutrients and removing waste products. Your brain's blood vessels, if stretched out end to end, would span about 400 miles. These delicate vessels form an intricate network that ensures every region of your brain receives the oxygen and nutrients it needs. The blood-brain barrier, a selective membrane, protects your brain from harmful substances while allowing necessary molecules to pass through. This barrier consists of tightly packed cells that form a protective shield around your brain tissue. Let your breathing slow as you contemplate the brain's division into 2 hemispheres, connected by the corpus callosum, a bundle of approximately 200 million nerve fibers. These hemispheres work together, though each has some specialized functions. The left hemisphere typically handles language processing, mathematical reasoning, and logical analysis in most people. The right hemisphere often specializes in spatial awareness, facial recognition, and creative thinking. This lateralization, however, is not absolute, and both hemispheres collaborate constantly through their interconnecting pathways. The frontal lobes, located behind your forehead, govern executive functions like planning, decision-making, and impulse control. These areas of the brain continue developing into your mid-20s, gradually refining your ability to make complex decisions and regulate emotions. The prefrontal cortex, the most forward portion of the frontal lobes, acts like an orchestra conductor, coordinating information from other brain regions to guide goal-directed behavior. Damage to this area can profoundly affect personality and judgment, demonstrating how specific brain regions contribute to who you are. Behind the frontal lobes lie the parietal lobes, which process sensory information from your body and help you navigate space. These regions integrate touch, temperature, and pain signals, creating your sense of where your body exists in the world. The parietal lobes also play crucial roles in mathematical reasoning and spatial manipulation, allowing you to rotate objects mentally or estimate distances. As you sink deeper into relaxation, your parietal lobes continue monitoring your body's position, maintaining awareness even as conscious thought fades. The temporal lobes, positioned near your temples, house the primary auditory cortex and play essential roles in memory formation. The hippocampus, a seahorse-shaped structure deep within the temporal lobe, acts as a memory consolidator, transforming short-term memories into long-term storage. During sleep, your hippocampus replays experiences from your day, strengthening neural connections and integrating new information with existing knowledge. This nightly memory consolidation occurs most actively during slow-wave sleep, the deepest stage of non-REM sleep. The occipital lobes at the back of your brain process visual information, though you see with your brain far more than with your eyes. Your eyes capture light and convert it to electrical signals, but your occipital cortex interprets these signals, constructing the rich visual experience of the world around you. Specialized areas within the occipital lobes detect motion, recognize faces, and perceive colors. Even in darkness, as you rest now, these visual processing areas remain active, generating the imagery of dreams. Deep within your brain, the limbic system orchestrates emotions and motivations. The amygdala, an almond-shaped structure, processes fear and emotional significance, helping you respond quickly to threats. The hippocampus works alongside the amygdala, ensuring that emotionally significant events become firmly encoded in memory. The thalamus, sometimes called the brain's relay station, directs sensory information to appropriate cortical areas for processing. Almost all sensory information passes through the thalamus before reaching conscious awareness, except for smell, which travels directly to olfactory processing centers. The hypothalamus, though small, regulates fundamental drives and bodily functions. This tiny region controls hunger, thirst, body temperature, and circadian rhythms. It communicates with the pituitary gland, directing the release of hormones that influence growth, metabolism, and stress responses. Right now, as you prepare for sleep, your hypothalamus coordinates with other brain regions to reduce alertness and lower your body temperature, creating optimal conditions for rest. The brainstem, connecting your brain to your spinal cord, maintains essential life functions without requiring conscious thought. The medulla oblongata controls breathing and heart rate, ensuring these vital processes continue automatically. The pons participates in sleep regulation and facial movements, while the midbrain coordinates eye movements and processes auditory and visual information. These ancient structures, evolutionarily older than the cortex, keep you alive moment by moment. The cerebellum, positioned at the brain's base beneath the occipital lobes, coordinates movement and balance. This structure contains more neurons than all other brain regions combined, packed into a compact space. The cerebellum refines motor commands, ensuring smooth, coordinated movements. It also plays surprising roles in cognitive functions, including attention, language processing, and emotional regulation. When you learn a new physical skill, your cerebellum gradually assumes control, allowing movements to become automatic and fluid. Neurons communicate through neurotransmitters, chemical messengers that cross synaptic gaps between cells. Dopamine, associated with reward and motivation, influences movement, learning, and pleasure. Serotonin affects mood, sleep, and appetite, with many antidepressant medications targeting serotonin systems. Norepinephrine enhances attention and arousal, preparing your body for action. GABA, the brain's primary inhibitory neurotransmitter, reduces neural activity and promotes calm. Glutamate, conversely, excites neurons and plays crucial roles in learning and memory. As you drift deeper, know that your brain generates electrical rhythms measured as brain waves. Beta waves, ranging from 13 to 30 Hz, characterize active, alert states. Alpha waves at 8 to 12 Hz appear during relaxed wakefulness. Theta waves, 4 to 7 Hz, emerge during light sleep and deep meditation. Delta waves, below 4 Hz, dominate during deep sleep. These rhythms reflect synchronized neural activity across vast populations of neurons. Sleep itself progresses through distinct stages, each serving specific purposes. Stage 1, the transition into sleep, lasts only a few minutes. Stage 2 features sleep spindles and K-complexes, distinctive wave patterns that may protect sleep from disruption. Stages 3 and 4, deep slow-wave sleep, promote physical restoration and memory consolidation. REM sleep, characterized by rapid eye movements, hosts vivid dreams and consolidates emotional memories and procedural skills. Your brain creates consciousness through mechanisms still not fully understood. The neural correlates of consciousness, patterns of brain activity associated with awareness, involve widespread cortical networks. The thalamus and cortex engage in reciprocal communication, generating the unified experience of awareness. Some theories suggest consciousness emerges from integrated information across brain regions, while others propose quantum processes in neural microtubules. The mystery of how physical brain activity produces subjective experience remains one of neuroscience's deepest questions. Memory takes multiple forms in your brain. Working memory, managed by the prefrontal cortex, holds information temporarily for immediate use. Semantic memory stores facts and concepts without contextual details about when you learned them. Episodic memory preserves personal experiences with rich contextual information. Procedural memory, managed by the basal ganglia and cerebellum, guides skills and habits, operating largely outside conscious awareness. These memory systems work together, though they rely on different neural circuits. The brain demonstrates remarkable plasticity, continuously reorganizing throughout life. When you learn something new, synaptic connections strengthen through a process called long-term potentiation. Neurons that fire together wire together, as repeated activation strengthens their connections. Conversely, unused connections weaken and may disappear, a process called synaptic pruning. This neuroplasticity allows recovery from brain injuries, as healthy regions sometimes compensate for damaged areas. Neurogenesis, the birth of new neurons, continues in certain brain regions throughout adulthood. The hippocampus and olfactory bulb generate new neurons that integrate into existing circuits. Exercise, learning, and enriched environments promote neurogenesis, while chronic stress suppresses it. These new neurons may contribute to memory formation and emotional regulation, though their precise roles remain under investigation. The brain's white matter, composed of myelinated axons, facilitates communication between regions. Myelin, a fatty insulation, wraps around axons like electrical wire insulation, dramatically increasing signal transmission speed. White matter continues developing into your 30s, gradually improving cognitive efficiency. Damage to white matter can disconnect brain regions, impairing coordination between different processing areas. Your brain processes faces using specialized circuits in the fusiform gyrus. This region, sometimes called the fusiform face area, responds more strongly to faces than to other objects. Humans possess an extraordinary ability to recognize thousands of faces and detect subtle expressions. Damage to face processing areas can cause prosopagnosia, or face blindness, where affected individuals cannot recognize even familiar faces. Language processing typically involves the left hemisphere's Broca's area and Wernicke's area. Broca's area, in the frontal lobe, manages speech production and grammatical processing. Wernicke's area, in the temporal lobe, handles language comprehension. These regions connect through a bundle of nerve fibers called the arcuate fasciculus. Damage to Broca's area produces effortful, telegraphic speech with preserved comprehension. Damage to Wernicke's area creates fluent but meaningless speech with impaired understanding. The brain perceives time through distributed networks rather than a single clock. The cerebellum and basal ganglia process durations from milliseconds to seconds, crucial for motor timing. The prefrontal cortex and hippocampus handle longer intervals, supporting memory and planning. Time perception varies with attention, emotion, and age, demonstrating its constructed nature. When you focus intensely, time seems to pass quickly, while anxiety can make minutes feel like hours. Pain processing involves multiple brain regions working together. The sensory cortex identifies pain location and intensity. The anterior cingulate cortex processes pain's emotional aspects. The prefrontal cortex modulates pain through attention and expectation. Chronic pain can reorganize brain networks, sometimes persisting even after tissue healing. This demonstrates how pain reflects brain activity rather than simply damaged tissue. The brain maintains body awareness through the insular cortex, which integrates signals from internal organs. This interoception provides feelings of hunger, thirst, heartbeat, and breath. The insula contributes to emotional awareness, as many emotions involve bodily sensations. Mindfulness practices enhance insular activity, potentially improving body awareness and emotional regulation. Mirror neurons, discovered in the motor cortex, activate both when performing actions and when observing others perform them. These neurons may support imitation learning, empathy, and social cognition. The mirror neuron system connects observation and action, allowing you to understand others' intentions by simulating their movements internally. Some researchers suggest mirror neuron dysfunction might contribute to autism spectrum disorders, though this remains controversial. The default mode network activates during rest and internally focused thought. This network, including the medial prefrontal cortex and posterior cingulate cortex, supports self-referential thinking, memory retrieval, and imagination. The default network deactivates during focused external tasks, but resumes activity during mind wandering. Its activity patterns may relate to creativity and spontaneous problem solving. Attention involves multiple neural systems. The dorsal attention network, including frontal and parietal regions, directs voluntary attention toward chosen targets. The ventral attention network, right lateralized, detects unexpected stimuli and reorients attention. The salience network, involving the anterior insula and anterior cingulate cortex, determines which stimuli deserve attention. These networks interact to balance goal-directed focus and environmental monitoring. The brain's reward system centers on dopamine pathways from the ventral tegmental area to the nucleus accumbens and prefrontal cortex. This circuit reinforces behaviors leading to positive outcomes, supporting learning and motivation. Addictive substances hijack these pathways, producing artificially strong reward signals. Natural rewards like food, social connection, and achievement activate these circuits more moderately. Stress activates the hypothalamic pituitary adrenal axis, releasing cortisol to mobilize energy resources. Acute stress enhances certain cognitive functions, improving focus and memory consolidation. Chronic stress, however, damages the hippocampus and impairs memory formation. Stress also affects the amygdala, potentially increasing anxiety and fear responses. Managing stress protects brain health and cognitive function. The brain processes music through distributed networks spanning both hemispheres. The auditory cortex analyzes pitch, rhythm, and timbre. The motor cortex activates during rhythm processing, even without movement. The limbic system responds to music's emotional content. Music training enhances brain plasticity, strengthening connections between auditory, motor, and cognitive regions. Musicians often show increased corpus callosum size, reflecting enhanced interhemispheric communication. Sleep deprivation impairs multiple cognitive functions. Attention and working memory decline after even one night of poor sleep. The prefrontal cortex becomes less active, reducing executive control. The amygdala becomes hyperactive, intensifying emotional reactions. Chronic sleep loss accelerates cognitive aging and may increase dementia risk. Sleep allows your brain to clear metabolic waste through the glymphatic system, particularly active during deep sleep. The brain constructs your sense of self through autobiographical memory and self-referential processing. The medial prefrontal cortex activates when thinking about yourself. The posterior cingulate cortex integrates self-relevant information across time. These regions create continuity of identity despite constant neural changes. Damage to these areas can alter personality and self-awareness, demonstrating the brain basis of selfhood. Decision-making involves evaluating options and predicting outcomes. The orbitofrontal cortex assigns values to choices based on expected rewards and punishments. The dorsolateral prefrontal cortex maintains goals and resists impulses. The anterior cingulate cortex detects conflicts between options. These regions work together, though emotion and reason interact rather than operate separately. Purely rational decisions prove impossible, as emotion provides value signals guiding choice. The brain develops in a predictable sequence from back to front. Sensory and motor regions mature first, followed by association areas integrating information across modalities. The prefrontal cortex matures last, continuing development into the mid-20s. This extended development allows extensive learning, but creates vulnerability during adolescence, when reward-seeking develops before full impulse control. Aging affects the brain progressively. Processing speed declines as white matter integrity decreases. The prefrontal cortex and hippocampus show gradual volume loss. However, accumulated knowledge and wisdom can compensate for processing changes. Cognitive reserve, built through education and mental stimulation, protects against age-related decline. Remaining mentally and physically active preserves brain health throughout life. The brain consumes glucose as its primary fuel, requiring approximately 120 grams daily. Unlike muscles, the brain cannot store significant glucose, depending on constant blood delivery. When glucose runs low, concentration and mood suffer. Ketones can provide alternative fuel during fasting, potentially offering neuroprotective benefits. Maintaining stable blood sugar supports optimal brain function. The blood-brain barrier protects neural tissue but complicates drug delivery. This selective barrier blocks most large molecules and many drugs. Small, lipid-soluble molecules cross more easily. The barrier can break down in disease or injury, sometimes allowing harmful substances to reach brain tissue. Understanding this barrier helps scientists design better neurological medications. Neurotransmitter balance affects mental health profoundly. Depression often involves reduced serotonin, norepinephrine, or dopamine signaling. Anxiety disorders may reflect imbalanced GABA and glutamate. Schizophrenia involves dysregulated dopamine systems. While chemical imbalances alone cannot explain mental illness, neurotransmitter systems clearly influence mood, thought, and behavior. The brain generates heat through constant metabolic activity. Blood flow helps dissipate this heat, preventing damage from overheating. Fever raises brain temperature, sometimes causing confusion or delirium. The brain's temperature regulation systems work precisely to maintain optimal function. Glial cells, once considered mere support cells, actively participate in brain function. Astrocytes regulate neurotransmitter levels and support the blood-brain barrier. Oligodendrocytes produce myelin in the central nervous system. Microglia act as immune cells, protecting against infection and clearing debris. Glial cells outnumber neurons and play essential roles in brain health. The brain coordinates voluntary movement through a hierarchy of control. The motor cortex sends commands to spinal motor neurons. The basal ganglia select and initiate movements while suppressing unwanted actions. The cerebellum refines movements, ensuring accuracy and smoothness. These systems work seamlessly, producing fluid, purposeful actions. Proprioception, your sense of body position, relies on sensors in muscles and joints sending information to the brain. The parietal cortex integrates these signals, creating body awareness in space. Proprioceptive impairment makes coordinated movement difficult, demonstrating how the brain constructs even basic movement awareness. The brain responds to exercise with increased blood flow and neurotrophin production. Brain-derived neurotrophic factor supports neuron survival and growth. Exercise promotes hippocampal neurogenesis and may protect against cognitive decline. Physical activity benefits the brain as much as the body. Meditation produces measurable brain changes. Regular practice increases gray matter density in regions supporting attention and emotional regulation. The default mode network shows altered activity, potentially reducing self-focused rumination. Meditation may slow age-related brain atrophy, though more research is needed. The brain processes social information through specialized networks. The temporoparietal junction helps understand others' mental states. The superior temporal sulcus analyzes biological motion and social cues. The medial prefrontal cortex supports thinking about others' beliefs and intentions. These regions enable navigation of complex social worlds. Creativity involves widespread brain networks rather than localized regions. The default mode network generates spontaneous ideas. The executive control network evaluates and refines them. Creative thinking requires both networks working together, balancing spontaneous generation with critical evaluation. The brain extracts statistical regularities from experience, learning probabilistic relationships unconsciously. This statistical learning happens automatically in language acquisition, visual perception, and motor control. The brain builds internal models of the world based on experience patterns. Synesthesia, where stimuli trigger unexpected sensory experiences, reveals brain organization. Some people perceive letters as inherently colored or taste shapes. Increased neural crosstalk between sensory regions may cause these experiences. Synesthesia demonstrates individual variation in brain wiring. The brain maintains multiple maps of the body. The primary sensory cortex contains a distorted body map with enlarged hands and lips, reflecting their sensory importance. The motor cortex maintains a similar motor map. These maps can reorganize after injury or training, demonstrating plasticity. Hunger and satiety signals involve the hypothalamus monitoring blood glucose, leptin, and ghrelin. These hormones communicate nutritional state to the brain. The hypothalamus coordinates appropriate responses, stimulating or suppressing appetite. Disrupted hunger regulation contributes to eating disorders and obesity. The brain's energy efficiency surpasses any computer, performing vast computations using only about 20 watts. Neurons operate near thermal noise limits, maximizing information processing per energy unit. This efficiency results from billions of years of evolutionary optimization. Learning changes synaptic strength through molecular cascades activated by neural activity. Calcium ions entering neurons trigger protein synthesis and receptor insertion. These molecular changes stabilize strengthened connections, converting temporary activity patterns into lasting memories. The brain generates expectations about incoming sensory information, predicting what you will experience. Prediction errors, mismatches between expectation and reality, drive learning and attention. The brain constantly updates its models based on these errors, refining its understanding of the world. Consciousness may emerge from integrated information across brain regions. The amount of integrated information, rather than total activity, may determine consciousness level. This theory suggests consciousness reflects how much a system is more than the sum of its parts. The pineal gland produces melatonin in response to darkness, promoting sleep. Light exposure, particularly blue wavelengths, suppresses melatonin production. Modern artificial lighting can disrupt this system, delaying sleep onset. The pineal gland translates environmental light cycles into biochemical signals the brain can use. The brain's immune system differs from the body's, with microglia providing primary defense. These cells constantly survey brain tissue, responding rapidly to injury or infection. Overactive microglia may contribute to neurodegenerative diseases, highlighting the importance of balanced immune responses. Synaptic transmission occurs in milliseconds, but building new memories takes hours. Protein synthesis during this consolidation period remains vulnerable to disruption. Sleep protects consolidation, which is why learning benefits from subsequent rest. The brain processes beauty through reward and emotion circuits. The orbitofrontal cortex responds to beautiful faces, art, and music. Individual aesthetic preferences vary, but certain patterns activate these circuits reliably across people. Beauty perception combines sensory processing with emotional and reward systems. Navigation relies on specialized brain systems. Place cells in the hippocampus fire when you occupy specific locations. Grid cells in the entorhinal cortex create coordinate systems for spatial mapping. Head direction cells track which way you face. These cells work together, allowing you to know where you are and navigate effectively. The brain maintains stable perception despite constant eye movements. Each time you move your eyes, the visual scene shifts across your retina. The brain compensates for these movements, maintaining perceptual stability. This demonstrates how perception reflects active brain construction rather than passive reception. Emotional memories often feel more vivid than neutral ones. The amygdala modulates memory consolidation based on emotional significance. Stress hormones released during emotional events enhance hippocampal encoding. This mechanism ensures you remember important experiences, but can create intrusive memories in trauma. The brain's flexibility allows recovery from early injuries. Young brains show remarkable reorganization when damaged, with healthy regions assuming functions of injured areas. This plasticity decreases with age but never disappears entirely. Rehabilitation exploits remaining plasticity to restore lost functions. Phantom limb sensations occur when the brain's body map persists after amputation. The cortical region for the missing limb remains active, sometimes creating vivid sensations. Mirror therapy can reduce phantom pain by providing visual feedback that satisfies the brain's expectations. The brain distinguishes self-generated sensations from external stimuli. This ability prevents you from tickling yourself, but allows you to feel when others touch you. The cerebellum may suppress predicted sensory consequences of your own actions. Expertise reorganizes brain networks. Extensive practice creates efficient, specialized circuits. London taxi drivers who memorize the city's layout show enlarged hippocampi. Musicians develop enhanced auditory motor connections. These changes demonstrate how experience literally reshapes the brain. The brain tolerates ambiguity poorly, often resolving unclear stimuli into definite perceptions. Bistable images, which support multiple interpretations, cause the brain to flip between alternatives. These perceptual switches occur spontaneously, revealing how the brain actively constructs coherent experiences. Social rejection activates brain regions processing physical pain. The anterior cingulate cortex responds to both pain types, suggesting shared mechanisms. This overlap may explain why social pain feels genuinely painful rather than merely metaphorically so. The brain processes numbers through regions near those handling fingers. This proximity may reflect how humans first learned mathematics through counting on fingers. Mathematical expertise develops additional specialized circuits but builds on these foundational systems. Trust and social bonding involve oxytocin, a hormone affecting brain circuits. The hypothalamus produces oxytocin, which influences the amygdala and other limbic regions. Oxytocin release during positive social interactions strengthens bonds and promotes cooperation. The brain creates three-dimensional perception from two-dimensional retinal images. Binocular disparity, the slight difference between each eye's view, provides depth information. Motion parallax, how objects move at different speeds when you move, also signals depth. The visual cortex integrates these cues, constructing spatial understanding. Fear learning occurs rapidly, but extinction requires repeated safe experiences. The amygdala forms fear associations quickly, promoting survival. The prefrontal cortex gradually inhibits these responses through extinction learning. Impaired extinction may contribute to anxiety disorders. The brain synchronizes activity across regions through oscillating rhythms. Gamma oscillations, around 40 Hz, may bind features into unified perceptions. Theta rhythms coordinate hippocampal activity during memory formation. These oscillations allow distributed regions to communicate effectively. Placebo effects demonstrate the brain's power over bodily states. Expectation of improvement activates endogenous pain relief systems. The prefrontal cortex and midbrain regions release endorphins, reducing pain. Placebos show measurable effects in brain imaging, revealing genuine neurobiological changes. The brain rarely operates at full capacity simultaneously. Different regions activate for different tasks, with most areas quiet at any moment. This selective activation conserves energy and prevents interference between competing processes. Intuition may reflect implicit processing in brain regions outside conscious awareness. The basal ganglia and other subcortical structures learn patterns automatically. These systems can guide decisions before conscious analysis, creating gut feelings about correct choices. The brain maintains multiple timescales simultaneously. Neurons fire in milliseconds, but learning spans hours to years. Brain development unfolds over decades. These nested timescales allow both rapid responses and gradual adaptation. Rest now in the knowledge that your brain continues its essential work throughout the night, consolidating memories, clearing waste, and preparing for tomorrow's experiences. This remarkable organ, with its billions of neurons and trillions of connections, operates tirelessly to create your experience of being alive. As you sleep, neural networks strengthen important connections and prune unused ones, optimizing your cognitive abilities. The brain's complexity extends to the molecular level, where countless proteins and molecules orchestrate neural function. Ion channels embedded in neuronal membranes control electrical signals by allowing sodium, potassium, calcium, and chloride ions to flow across cell boundaries. These channels open and close in response to voltage changes or chemical signals, generating the action potentials that propagate along axons. The sodium-potassium pump continuously maintains concentration gradients, using energy from ATP to pump sodium out and potassium in, keeping neurons ready to fire. This molecular machinery operates with astonishing precision, timing events in microseconds and maintaining gradients across membranes only a few nanometers thick. Neurotransmitter receptors come in two main types, ionotropic and metabotropic. Ionotropic receptors contain ion channels that open directly when neurotransmitters bind, producing fast synaptic responses within milliseconds. Metabotropic receptors activate intracellular signaling cascades through G proteins, producing slower but longer-lasting effects. A single neurotransmitter can bind to multiple receptor types, producing different effects depending on receptor distribution. Glutamate, for example, binds to both AMPA receptors for fast excitation and NMDA receptors that require both glutamate and membrane depolarization, making them coincidence detectors crucial for learning. The brain maintains precise chemical environments in different regions and compartments. The extracellular fluid surrounding neurons differs from intracellular fluid and blood plasma in ion concentrations and pH. Astrocytes regulate this extracellular environment, removing excess neurotransmitters and potassium ions. They also provide neurons with lactate, an energy substrate produced from glucose. This metabolic coupling between astrocytes and neurons supports the brain's high-energy demands. Brain metabolism produces reactive oxygen species as byproducts, potentially damaging cells. The brain contains antioxidant systems, including superoxide dismutase, catalase, and glutathione, that neutralize these harmful molecules. Mitochondria, the cellular powerhouses, generate most of the brain's energy through oxidative phosphorylation. Each neuron contains hundreds to thousands of mitochondria, particularly concentrated in synapses where energy demands peak. Mitochondrial dysfunction contributes to neurodegenerative diseases, highlighting the importance of cellular energy production for brain health. The cytoskeleton, an internal scaffold of protein filaments, maintains neuronal structure and enables transport. Microtubules extend from the cell body through axons and dendrites, serving as tracks for motor proteins that carry cargo. Kaizen motors move materials toward axon terminals, while dynein motors transport materials back to the cell body. This bidirectional transport delivers neurotransmitter vesicles, organelles, and proteins, where needed, while returning damaged components for recycling. Disrupted axonal transport contributes to several neurological disorders. Synaptic vesicles containing neurotransmitters cluster near release sites in axon terminals. When an action potential arrives, voltage-gated calcium channels open, allowing calcium ions to flood into the terminal. Calcium triggers snare proteins to pull vesicles against the cell membrane, causing fusion and neurotransmitter release. This process, called exocytosis, occurs with remarkable speed and precision. Empty vesicles recycle through endocytosis, refilling with neurotransmitter for subsequent releases. A single synapse can recycle vesicles hundreds of times per second during intense activity. The postsynaptic density, a protein complex beneath the postsynaptic membrane, anchors receptors and organizes signaling molecules. This structure contains hundreds of different proteins, forming a sophisticated molecular machine. Receptor trafficking constantly adjusts synapse strength, with receptors inserted during potentiation and removed during depression. This dynamic regulation allows synapses to strengthen or weaken based on activity patterns, implementing the cellular mechanisms of learning and memory. Dendritic spines, tiny protrusions from dendrites, house most excitatory synapses. These spines change shape dynamically, growing larger with stimulation and shrinking or disappearing with inactivity. Spine growth requires actin polymerization, building a cytoskeletal framework that enlarges the spine head. This structural plasticity accompanies functional changes in synaptic strength, providing a physical substrate for memory storage. The brain contains billions of these tiny spines, each potentially representing a memory trace. Myelination dramatically increases conduction speed by causing action potentials to jump between nodes of Ranvier, gaps in the myelin sheath. This saltatory conduction allows signals to travel up to 100 times faster than in unmyelinated axons. Multiple sclerosis, an autoimmune disease that damages myelin, demonstrates the importance of this insulation. As myelin degrades, signals slow or fail to propagate, causing diverse neurological symptoms depending on lesion locations. The brain exhibits critical period plasticity, heightened sensitivity to experience during specific developmental windows. Visual cortex development requires patterned visual input during early life. Deprivation during this critical period causes permanent deficits, even if vision is restored later. Language acquisition similarly shows critical period effects, with second languages learned after puberty typically showing different neural organization than first languages. These windows reflect molecular mechanisms that make young brains especially plastic. Epigenetic modifications alter gene expression without changing DNA sequences. Methyl groups attached to DNA or histones can silence genes, while acetyl groups typically activate them. Experience drives epigenetic changes that potentially last lifetimes. Early stress can produce lasting alterations in stress response genes through epigenetic mechanisms. These modifications potentially transmit across generations, though this remains controversial. Epigenetics provides a mechanism for the environment to leave lasting marks on the genome. The brain's immune response differs from peripheral immunity due to its limited regenerative capacity. Microglia respond to injury by proliferating and adopting activated states. Some activation states support repair, clearing debris and releasing growth factors. Other states promote inflammation that can damage healthy tissue. Controlling microglial activation might help treat neurodegenerative diseases where chronic inflammation contributes to neuronal loss. Brain oscillations coordinate activity across distributed neurons. Slow oscillations during deep sleep may facilitate synaptic downscaling, reducing overall synaptic strength and maintaining neural homeostasis. Sleep spindles, brief bursts of activity, may protect sleep from disruption while facilitating memory consolidation. Theta oscillations pace hippocampal activity during exploration and memory encoding. These rhythms organize neural firing, allowing temporal coordination of activity across brain regions. The brain predicts sensory consequences of action, suppressing expected sensations. This forward model, likely involving the cerebellum and parietal cortex, explains why self-produced sensations feel attenuated. When predictions fail to match sensory input, prediction errors alert you to unexpected events. This predictive processing framework suggests perception, reflects the brain's best guess about causes of sensory inputs, rather than passive registration of stimuli. The claustrum, a thin sheet of neurons beneath the cortex, connects extensively with cortical regions. Its function remains mysterious, but dense interconnections suggest integration roles. Some theories propose the claustrum coordinates information across cortical areas, possibly contributing to consciousness. Its unusual anatomy and connectivity make it an intriguing target for neuroscience research. Neuronal diversity far exceeds simple categories like excitatory and inhibitory. Interneurons, comprising about 20% of cortical neurons, include dozens of distinct subtypes with unique properties. Parvalbumin-expressing interneurons provide fast, powerful inhibition, synchronizing neural populations. Somatostatin-expressing interneurons target dendrites, gating synaptic inputs. VIP-expressing interneurons preferentially inhibit other interneurons, disinhibiting principal neurons. This interneuron diversity enables sophisticated circuit computations. The reticular activating system, a network in the brainstem, regulates arousal and consciousness. Damage to this system causes coma, demonstrating its essential role in wakefulness. The RAS receives sensory information and projects widely to cortex, thalamus, and hypothalamus, modulating overall brain activity levels. Neuromodulators from the RAS, including norepinephrine, serotonin, dopamine, and acetylcholine, adjust cortical processing according to behavioral state. Acetylcholine enhances attention and learning by modulating cortical circuits. Cholinergic projections from the basal forebrain increase cortical plasticity and responsiveness. During sleep, acetylcholine levels drop in most brain regions except during REM sleep when they approach waking levels. This REM acetylcholine may support memory consolidation and dream generation. Alzheimer's disease involves early degeneration of cholinergic neurons, contributing to memory impairment. The locus coeruleus, a small brainstem nucleus, projects norepinephrine throughout the brain. This system activates during stress, novelty, and effort, enhancing arousal and focus. The locus coeruleus also influences memory consolidation, with norepinephrine release strengthening emotionally significant memories. This nucleus degenerates early in Alzheimer's disease and may accumulate abnormal proteins that spread to other brain regions. Serotonergic neurons in the raphe nuclei influence mood, sleep, and impulse control. These neurons fire regularly during waking, slow during slow-wave sleep, and nearly cease during REM sleep. Selective serotonin reuptake inhibitors increase synaptic serotonin by blocking reuptake transporters. Despite widespread use, the mechanisms by which SSRIs alleviate depression remain incompletely understood, likely involving changes in neural circuits beyond simple neurotransmitter levels. Dopaminergic neurons in the substantia nigra project to the striatum, controlling movement initiation. Parkinson's disease results from degeneration of these neurons, producing characteristic motor symptoms. Dopamine replacement with L-Dopa, a precursor that crosses the blood-brain barrier, dramatically relieves symptoms initially. However, long-term treatment complications arise, potentially involving maladaptive plasticity in dopamine-deprived circuits. The ventral tegmental area sends dopamine to limbic regions, mediating reward and motivation. This mesolimbic pathway reinforces behaviors leading to rewards. Addictive drugs hijack this system, producing dopamine surges that dwarf natural rewards. Repeated drug use causes adaptations that reduce reward sensitivity, requiring larger doses for equivalent effects while diminishing pleasure from natural rewards. The habenula, a small epithalamic structure, signals negative outcomes and inhibits dopamine neurons. This anti-reward center may contribute to depression when overactive. The habenula influences decisions to avoid punishment or withhold effort when rewards seem unlikely. Its role in motivated behavior makes it an intriguing target for depression research. The amygdala contains multiple nuclei with distinct functions. The lateral nucleus receives sensory information and forms fear associations. The central nucleus outputs to brainstem regions controlling fear responses like freezing and increased heart rate. The basal nucleus connects to prefrontal cortex, supporting fear extinction. This internal organization allows the amygdala to acquire, express, and regulate emotional responses. The bed nucleus of the stria terminalis, closely related to the amygdala, mediates sustained anxiety rather than acute fear. While the amygdala responds to immediate threats, the BNSD activates during prolonged stress or uncertainty. These parallel systems handle threats on different timescales, with the BNSD contributing to anxiety disorders. The hippocampus consists of several subregions with specialized roles. The dentate gyrus receives inputs from entorhinal cortex and performs pattern separation, creating distinct representations of similar experiences. CA3 performs pattern completion, retrieving full memories from partial cues. CA1 compares expected and actual inputs, potentially signaling prediction errors. These computations enable episodic memory formation and spatial navigation. The entorhinal cortex provides the hippocampus with sensory information and contains grid cells that tile the environment with hexagonal fields. Grid cells, along with place cells, head direction cells, and border cells, create a neural map supporting spatial memory. These specialized neurons won the discoverers the Nobel Prize, revealing how the brain represents space at the cellular level. The parahynal cortex processes object information, complementing the hippocampus's spatial processing. Damage to this region impairs object recognition while sparing spatial memory. The combined system of medial temporal lobe structures creates memories that bind objects, places, and contexts into coherent episodes. Reconsolidation reveals memory's dynamic nature. Each time you recall a memory, it becomes temporarily unstable before reconsolidating. This window allows memories to update with new information or weaken through interference. Reconsolidation-based therapies might eventually treat PTSD by weakening traumatic memories during retrieval. The striatum receives inputs from throughout the cortex and participates in action selection and habit formation. The dorsal striatum develops habitual responses through repeated practice. The ventral striatum evaluates outcomes and motivates behavior. Direct and indirect pathways through the striatum facilitate selected actions while suppressing alternatives, implementing a neural mechanism for decision-making. The globus pallidus and subthalamic nucleus, parts of the basal ganglia, refine movement selection through inhibitory and excitatory loops. Deep brain stimulation of the subthalamic nucleus treats Parkinson's symptoms by modulating these circuits. The precise mechanisms remain debated, but stimulation clearly reorganizes pathological activity patterns. The superior colliculus in the midbrain directs eye movements and attention shifts. This structure creates a map of visual space and generates commands to orient eyes, head, and attention toward targets. The superior colliculus receives direct retinal input, enabling rapid reflexive saccades even without cortical involvement. The inferior colliculus processes auditory information before relaying to the thalamus. This midbrain structure extracts sound features and participates in sound localization. The inferior colliculus contains a tonotopic map with different frequencies represented in organized spatial arrays. The periaqueductal gray surrounds the cerebral aqueduct in the midbrain and organizes defensive behaviors. Electrical stimulation of different PAG columns produces distinct behaviors like freezing, flight, or fight. The PAG also modulates pain through descending control of spinal pain transmission. This ancient structure coordinates adaptive responses to threats. The red nucleus influences movement through the rubrospinal tract, though this pathway is less prominent in humans than other mammals. The red nucleus receives cerebellar input and may compensate partially for motor cortex damage. Its role in human motor control remains less understood than other motor structures. The ventral tegmental area not only releases dopamine, but also contains neurons releasing GABA and glutamate. These non-dopaminergic neurons participate in reward processing and aversion, revealing greater complexity than previously appreciated. VTA circuits integrate information about rewards, effort, and outcomes to guide motivated behavior. The locus coeruleus responds to salient stimuli with brief bursts of activity, releasing norepinephrine that enhances processing of important information. This system implements a neural interrupt signal, redirecting attention to significant events. Tonic locus coeruleus activity tracks arousal levels, while phasic bursts respond to specific stimuli. The tuberomammillary nucleus in the hypothalamus produces histamine, promoting wakefulness. Antihistamines that cross the blood-brain barrier cause drowsiness by blocking these arousal signals. The tuberomammillary nucleus receives input from the circadian clock, helping align sleep-wake cycles with day-night rhythms. The suprachiasmatic nucleus serves as the brain's master circadian clock. Light information reaches this hypothalamic nucleus directly from specialized retinal ganglion cells containing melanopsin. The SCN synchronizes peripheral clocks throughout the body, coordinating daily rhythms in physiology and behavior. Genetic mutations affecting clock genes can shift circadian phase, creating extreme morning or even an area postrema lack a complete blood-brain barrier, allowing direct detection of blood-borne signals. These circumventricular organs monitor circulating hormones and toxins, triggering appropriate responses. The area postrema induces vomiting when detecting toxins, while the subfornical organ regulates thirst and blood pressure. The choroid plexus produces cerebrospinal fluid, secreting about 500 milliliters daily. CSF cushions the brain, provides buoyancy that reduces effective brain weight and clears metabolic waste. The choroid plexus also expresses tight junctions forming the blood-CSF barrier, controlling which substances enter CSF. Cerebrospinal fluid circulates through ventricles and around the brain and spinal cord before draining into blood through arachnoid granulations. Recently discovered glymphatic clearance, most active during sleep, moves CSF through brain tissue along perivascular spaces. This system removes waste proteins including amyloid beta, which accumulates in Alzheimer's disease. The meninges, 3 membranes surrounding the brain, protect against injury and infection. The dura mater forms a tough outer layer. The arachnoid mater creates a space for CSF circulation. The pia mater follows brain contours closely, carrying blood vessels into brain tissue. Meningitis, inflammation of these membranes, can be life-threatening. The vertebral and carotid arteries supply blood to the brain through the circle of Willis, an arterial ring at the brain's base. This arrangement provides collateral circulation, allowing blood to reach brain regions through multiple routes. The circle of Willis varies anatomically between individuals, affecting stroke risk and outcomes. Brain capillaries form dense networks, placing every neuron within micrometers of a blood vessel. This proximity ensures rapid nutrient delivery and waste removal. Capillary density varies across regions, highest in metabolically demanding areas like the cortex and cerebellum. Angiogenesis, formation of new capillaries, can occur in response to increased activity or during recovery from injury. Neurovascular coupling adjusts blood flow to match local metabolic demands. Active neurons release signals causing nearby arterioles to dilate, increasing blood flow to active regions. This coupling underlies functional brain imaging techniques like fMRI, which detect blood oxygenation changes reflecting neural activity. The mechanisms ensuring tight neurovascular coupling remain actively investigated. Stroke occurs when blood flow interruption deprives brain tissue of oxygen and glucose. Ischemic strokes caused by clots account for most cases. Hemorrhagic strokes result from ruptured vessels. The brain tolerates only brief oxygen deprivation before neurons begin dying. Rapid treatment can minimize damage by restoring blood flow or removing clots. The penumbra, tissue surrounding a stroke's core, remains viable but functionally impaired. This tissue can recover if blood flow restores quickly, making rapid treatment critical. Neuroprotective strategies aim to extend the time penumbral tissue survives, widening the treatment window. Traumatic brain injury results from external forces, causing structural damage or functional disruption. Concussions, mild TBIs, may not involve obvious structural damage but temporarily disrupt brain function. Repeated concussions accumulate damage, potentially causing chronic traumatic encephalopathy. The brain's vulnerability to mechanical injury reflects its soft consistency and complex structure. Brain tumors can originate from glial cells, meningeal cells, or other brain components. Gliomas arising from glial cells range from slow-growing astrocytomas to aggressive glioblastomas. Brain tumors cause symptoms through mass effects, infiltration, or seizure induction. Treatment challenges include the blood-brain barrier limiting drug delivery and the need to preserve critical brain tissue. Epilepsy involves recurrent seizures resulting from excessive neural synchronization. Focal seizures originate in specific brain regions, while generalized seizures affect both hemispheres simultaneously. Antiepileptic drugs reduce seizure frequency by modulating ion channels or neurotransmitter systems. Some patients with drug-resistant epilepsy benefit from surgical removal of seizure foci. Seizures propagate through neural networks when excitation overwhelms inhibition. Excessive glutamate release, impaired GABA inhibition, or altered ion channel function can trigger seizures. During seizures, massive neuronal firing depletes energy stores and can cause excitotoxic damage. Understanding seizure mechanisms guides development of more effective treatments. Multiple sclerosis involves autoimmune attacks on myelin, disrupting neural communication. Symptoms vary depending on lesion locations and can include visual problems, weakness, sensory changes, and cognitive impairment. Disease-modifying therapies reduce attack frequency by suppressing immune system components targeting myelin. Repair of damaged myelin remains a major research goal. Alzheimer's disease causes progressive memory loss and cognitive decline. Amyloid plaques and tau tangles accumulate in vulnerable brain regions, particularly the hippocampus and association cortex. Neuroinflammation contributes to neuronal death. Current treatments provide modest symptomatic relief but do not halt disease progression. Recent antibody therapies targeting amyloid show promise for slowing cognitive decline. Parkinson's disease results from dopaminergic neuron loss in the substantia nigra. Motor symptoms including tremor, rigidity, and slowness reflect striatal dopamine depletion. Non-motor symptoms like depression, sleep disorders, and cognitive changes can precede motor symptoms. Lewy bodies containing misfolded alpha-synuclein protein characterize affected neurons. Understanding protein misfolding mechanisms might enable disease-modifying therapies. Huntington's disease, caused by a genetic mutation producing abnormal huntingtin protein, causes progressive motor, cognitive, and psychiatric symptoms. The mutation involves expanded CAG repeats in the huntingtin gene. Mutant huntingtin damages neurons, particularly in the striatum. Genetic testing allows presymptomatic diagnosis, raising ethical considerations about testing and disclosure. Amyotrophic lateral sclerosis causes progressive motor neuron degeneration, leading to paralysis and respiratory failure. Most cases arise sporadically, though familial forms involve genetic mutations. ALS demonstrates selective vulnerability, with motor neurons dying while nearby neurons survive. Understanding this selective vulnerability might reveal therapeutic targets. Autism spectrum disorders involve altered brain development affecting social communication and behavior. Genetic factors play significant roles, with hundreds of genes contributing to ASD risk. Brain differences include altered connectivity patterns and regional volume changes. early intervention improves outcomes, though no cure exists. The neurodiversity movement emphasizes autism as difference rather than purely deficit. Schizophrenia typically emerges in late adolescence or early adulthood with positive symptoms like hallucinations and delusions, negative symptoms like reduced motivation and emotional expression, and cognitive impairments. Antipsychotic medications primarily target dopamine receptors, reducing positive symptoms but addressing negative and cognitive symptoms less effectively. Brain changes include enlarged ventricles and reduced gray matter in several regions. Depression involves persistent low mood, anhedonia, and other symptoms affecting thinking, behavior, and physical health. Multiple brain systems show altered function, including reduced activity in dorsolateral prefrontal cortex and increased activity in default mode network. Antidepressants modulate monoamine neurotransmitters, but therapeutic effects take weeks, suggesting downstream changes in neuroplasticity rather than direct neurotransmitter effects cause improvement. Bipolar disorder alternates between depressive and manic episodes. During mania, elevated mood, increased energy, and impulsivity reflect altered prefrontal and limbic function. Mood stabilizers like lithium prevent episodes, possibly by enhancing neuroplasticity and neuroprotection. Brain imaging reveals altered activation in circuits regulating emotion and reward. Anxiety disorders involve excessive fear or worry causing significant distress. The amygdala shows heightened reactivity, while prefrontal regulatory regions show reduced activity. Cognitive behavioral therapy proves effective by strengthening prefrontal control over limbic responses. Exposure therapy, repeatedly confronting feared situations without harm, leverages extinction learning to reduce anxiety. Obsessive-compulsive disorder involves intrusive thoughts and repetitive behaviors aimed at reducing distress. Brain circuits connecting the orbitofrontal cortex, striatum, and thalamus show abnormal activity. SSRIs reduce symptoms in many patients, while exposure and response prevention therapy helps patients resist compulsions. OCD demonstrates how specific circuits generate distinct symptom profiles. Post-traumatic stress disorder develops after experiencing or witnessing trauma. PTSD involves intrusive memories, avoidance, negative thoughts and mood, and hyperarousal. The amygdala becomes hyperresponsive while the prefrontal cortex shows reduced activation. Effective treatments include trauma-focused therapies that help process traumatic memories in safe contexts. Sleep disorders disrupt normal sleep patterns with wide-ranging consequences. Insomnia involves difficulty initiating or maintaining sleep. Sleep apnea causes repeated breathing interruptions. Narcolepsy involves sudden sleep attacks and cataplexy. REM behavior disorder features acting out dreams due to loss of normal REM sleep paralysis. Each disorder reflects dysfunction in specific sleep regulatory circuits. The ascending arousal system promotes wakefulness through multiple neurotransmitters. Orexin neurons in the lateral hypothalamus stabilize wake-sleep transitions. Loss of orexin neurons causes narcolepsies, debilitating sleepiness and cataplexy. Orexin antagonists can treat insomnia by facilitating sleep initiation. Sleep pressure accumulates during waking through adenosine buildup. Adenosine, a byproduct of ATP metabolism, inhibits arousal-promoting neurons. Caffeine blocks adenosine receptors, preventing this inhibition and maintaining alertness. As adenosine clears during sleep, sleep pressure dissipates, allowing wakefulness to return. Dreams occur most vividly during REM sleep, though dreaming also occurs in non-REM stages. REM dreams tend to be bizarre and emotionally intense, reflecting activation patterns in limbic and visual areas. The prefrontal cortex's reduced activity, may contribute to dream illogic and poor memory for dreams. Dream functions remain debated, with theories proposing roles in emotional regulation, threat simulation, or random neural activity. Lucid dreaming, awareness of dreaming while dreaming, occurs spontaneously in some people and can be trained. During lucid dreams, prefrontal cortex activity increases compared to normal dreams, suggesting restored metacognitive awareness. Lucid dreamers can signal their state through planned eye movements, enabling laboratory study of this unique consciousness state. The brain exhibits sexual dimorphism, with structural and functional differences between sexes. These differences reflect hormonal influences during development and throughout life. Sex differences appear in spatial cognition, verbal abilities, and stress responses, though considerable overlap exists and environmental factors shape these traits significantly. Understanding sex differences helps address health disparities in neurological and psychiatric conditions. Hormones profoundly influence brain function. Estrogen affects hippocampal plasticity and memory, with some cognitive functions fluctuating across the menstrual cycle. Testosterone influences spatial cognition and aggression. Cortisol, released during stress, affects memory consolidation differently depending on stress timing. Thyroid hormones are essential for brain development, with deficiency causing irreversible intellectual disability if untreated in infancy. Pregnancy causes substantial brain changes. Gray matter volume decreases in regions supporting social cognition, possibly reflecting synaptic pruning that enhances maternal behavior. These changes persist for years postpartum. Pregnancy also affects memory and emotional processing. Understanding these neural adaptations illuminates how the brain supports caregiving behaviors. Aging affects the brain gradually, with some regions more vulnerable than others. Processing speed declines as white matter degrades. Episodic memory deteriorates while semantic knowledge often remains intact. However, cognitive reserve built through education and mental stimulation helps maintain function. Staying physically active, socially engaged, and mentally challenged supports brain health throughout aging. The gut-brain axis demonstrates bidirectional communication between the digestive system and brain. The vagus nerve carries signals from gut to brain, informing about nutritional state and microbial activity. Gut microbiota produce neurotransmitters and metabolites affecting brain function. Probiotic interventions may influence mood and cognition, though mechanisms remain unclear. This axis highlights how bodily systems influence mental states. Inflammation affects brain function and may contribute to depression and neurodegenerative diseases. Cytokines released during inflammation can alter neurotransmitter metabolism and neural activity. Chronic low-grade inflammation, increasingly common in modern environments, might increase psychiatric disorder risk. Anti-inflammatory intervention show promise for treating some patients with depression. Exercise benefits the brain through multiple mechanisms. Aerobic exercise increases hippocampal volume and improves memory. Exercise releases brain-derived neurotrophic factor, supporting neuronal health and plasticity. Physical activity reduces inflammation, improves vascular health, and may delay cognitive decline. Even modest regular exercise confers cognitive benefits. Diet influences brain health, with Mediterranean and MIND diets associated with reduced dementia risk. These diets emphasize vegetables, fruits, whole grains, fish, and healthy fats while limiting red meat and processed foods. Specific nutrients like omega-3 fatty acids support brain structure and function. The brain's nutritional requirements change across the lifespan, with different demands during development, adulthood, and aging. Hydration affects cognitive function, with even mild dehydration impairing attention and memory. The brain's high water content requires adequate hydration for optimal function. Electrolyte balance also matters, as neurons depend on sodium and potassium gradients. Maintaining proper hydration supports all brain functions. Sleep enhances learning and memory consolidation across domains. Motor skill learning benefits from sleep, with procedural memories strengthening offline. Emotional memory consolidation occurs during REM sleep. Problem solving improves after sleep, potentially reflecting unconscious processing. These benefits motivate prioritizing adequate sleep for optimal cognitive function. Meditation trains attention and emotional regulation through sustained practice. Focused attention meditation strengthens attention networks. Open monitoring meditation enhances awareness of thoughts and sensations without judgment. Loving-kindness meditation cultivates compassion. Different practices engage distinct neural circuits, offering diverse mental training approaches. Stress management protects brain health by reducing chronic cortisol exposure. Stress reduction techniques activate parasympathetic nervous system responses, lowering arousal. Regular relaxation practice may protect the hippocampus from stress-related damage. Social support buffers stress effects, demonstrating how relationships influence brain health. Music engages multiple brain systems simultaneously, from auditory processing to motor control to emotional responses. Musical training enhances auditory discrimination, motor skills, and executive functions. Music therapy helps patients with Parkinson's disease, stroke, and dementia. Music's powerful emotional effects reflect its ability to activate reward circuits and trigger memories. Language learning creates new neural connections and may provide cognitive reserve. Bilingual individuals show enhanced executive control, possibly from constantly managing multiple languages. Second language learning remains possible throughout life, though neural organization differs from native language acquisition. Language learning exercises memory, attention, and cognitive flexibility. Social interaction engages complex brain networks supporting theory of mind, empathy, and communication. Social connections benefit mental and physical health, with loneliness increasing disease risk. The brain evolved for social living, with extensive neural machinery devoted to navigating social worlds. Maintaining social relationships throughout life protects cognitive function. Creativity involves coordinated activity in imagination, memory, and cognitive control networks. Creative thinking requires both generating novel ideas and evaluating their value. The brain balances spontaneous generation with executive control during creative processes. Creativity training may enhance these abilities by strengthening relevant circuits. Flow states, characterized by complete absorption in activities, show distinctive brain patterns. Flow involves reduced default mode network activity and heightened focus. Time perception changes, and self-consciousness fades. Understanding flow's neural basis might enable intentionally cultivating this optimal experience state. The brain's remarkable plasticity means learning continues throughout life. Each experience potentially modifies neural connections, creating an ever-evolving organ. This plasticity underlies resilience, recovery from injury, and lifelong learning capacity. Your brain grows and changes with every thought, every memory, every moment of awareness. As you rest tonight, your brain continues its quiet work, processing the day's experiences, consolidating important information, and preparing for tomorrow. The vast complexity within your skull operates seamlessly, creating your experience of being alive, thinking, feeling, and dreaming. The brain demonstrates remarkable compensatory abilities when faced with challenges. After stroke, surviving neurons can sometimes assume functions of damaged areas through reorganization of neural networks. This compensation varies with lesion location, size, and individual factors. Rehabilitation therapies exploit remaining plasticity, encouraging beneficial reorganization through intensive practice. Constraint-induced therapy, forcing use of impaired limbs, promotes cortical reorganization supporting functional recovery. Brain-computer interfaces create direct connections between neural activity and external devices. These systems decode motor intentions from cortical activity, enabling paralyzed individuals to control cursors or robotic limbs. Current interfaces require surgical electrode implantation, but non-invasive approaches using EEG show promise. BCIs might eventually restore communication for locked-in patients or enhance human capabilities. The brain's computational principles inspire artificial neural networks used in machine learning. Artificial neurons receive weighted inputs, apply nonlinear functions, and produce outputs. Networks learn by adjusting connection weights based on performance feedback. Deep learning networks with many layers extract hierarchical features from data. However, artificial networks differ fundamentally from biological brains in architecture, learning rules, and efficiency. Optogenetics allows precise control of neural activity using light-sensitive proteins. Channel rhodopsin, activated by blue light, opens ion channels and excites neurons. Halorhodopsin, activated by yellow light, inhibits neurons. Expressing these proteins in specific cell types enables researchers to activate or silence those neurons with millisecond precision. This technology revolutionized neuroscience by allowing causal tests of neural circuit functions. Calcium imaging reveals activity in large neural populations simultaneously. Genetically encoded calcium indicators fluoresce when calcium enters active neurons. Two-photon microscopy images these indicators deep in living brain tissue. Researchers can record thousands of neurons while animals perform behaviors, revealing how neural populations encode information and coordinate actions. Functional MRI detects blood oxygenation changes reflecting neural activity. Active brain regions require increased blood flow, changing the magnetic resonance signal. fMRI provides whole brain coverage with millimeter spatial resolution, though temporal resolution lags behind neural activity by seconds. This technique maps brain regions supporting various cognitive functions. Electroencephalography records electrical activity from scalp electrodes, providing millisecond temporal resolution. EEG primarily detects synchronized activity in cortical pyramidal neurons oriented perpendicular to the scalp. Different frequency bands associate with distinct brain states. EEG remains valuable for diagnosing epilepsy, monitoring anesthesia depth, and studying sleep. Magnetoencephalography measures magnetic fields generated by neural activity. MEG provides better spatial resolution than EEG by detecting fields that pass through the skull undistorted. This technique excels at tracking rapid neural dynamics during perception and cognition. MEG requires magnetically shielded rooms and sensitive detectors, limiting availability. Positron emission tomography uses radioactive tracers to image brain chemistry and metabolism. PET can measure neurotransmitter receptors, glucose metabolism, or protein accumulation. Amyloid PET imaging detects Alzheimer's pathology in living patients. PET provides unique information unavailable from other imaging methods, but involves radiation exposure. Transcranial magnetic stimulation applies magnetic pulses that induce electrical currents in the brain. TMS can temporarily disrupt activity in targeted regions, testing their necessity for specific functions. Repetitive TMS modulates cortical excitability, with therapeutic applications in depression. TMS provides non-invasive tools for both research and treatment. Transcranial direct current stimulation passes weak electrical currents through the scalp, modulating neural excitability. Anodal stimulation typically enhances cortical activity, while cathodal stimulation reduces it. TDCS shows promise for enhancing learning, treating depression, and promoting stroke recovery. However, optimal parameters and mechanisms remain under investigation. The brain exhibits degeneracy, where multiple neural configurations can produce similar functions. This redundancy provides resilience against damage and enables diverse paths to behavioral goals. Degeneracy differs from simple redundancy, as the same elements participate in different functions in different contexts. This property allows flexible adaptation to changing demands. Neural Darwinism proposes that brain development involves selecting from overproduced neural connections. Initially, excessive synapses compete for stability based on activity patterns. Frequently used connections strengthen, while unused ones weaken and disappear. This selection process sculpts functional circuits matched to experienced environments. Experience thus shapes brain development through selective stabilization, rather than directing specific connections. The binding problem asks how the brain combines information processed in separate regions into unified perceptions. Color, shape, motion, and location are extracted by different visual areas, yet you perceive coherent objects. Temporal synchronization may bind features, with neurons representing the same object firing in synchrony. Attention might also contribute by selectively enhancing representations of attended objects. The hard problem of consciousness questions why neural activity produces subjective experience. Science explains many aspects of consciousness like wakefulness, attention, and information integration. However, explaining why these processes feel like anything remains mysterious. Some philosophers argue subjective experience cannot be reduced to physical processes, while others believe better understanding of brain function will illuminate consciousness. Free will and determinism raise profound questions about brain function and human agency. If neural activity determines decisions, and neural activity follows physical laws, then decisions seem predetermined. However, complex systems like brains exhibit emergent properties, not simply reducible to components. The brain's ability to represent counterfactuals and simulate consequences may provide a form of freedom even within a deterministic framework. The self emerges from brain processes integrating bodily signals, memories, and social information. The medial prefrontal cortex supports self-referential processing. The temporoparietal junction distinguishes self from other. The posterior cingulate cortex maintains narrative identity across time. These regions construct the experience of being a continuing self despite constant neural change. Attention selects information for detailed processing from the overwhelming sensory input. Bottom-up attention captures attention through stimulus salience. Top-down attention directs focus toward goals. Attention enhances processing of selected information while suppressing distractors. Attentional capacity limits mean you cannot fully process everything simultaneously, requiring strategic allocation of this precious resource. Working memory holds information temporarily for ongoing cognitive operations. The phonological loop maintains verbal information through rehearsal. The visuospatial sketchpad handles visual and spatial content. The episodic buffer integrates information across domains. Working memory capacity limits constrain complex thinking, though strategies can partially overcome these limits. Executive functions including planning, cognitive flexibility, and inhibitory control depend on prefrontal cortex. These abilities develop slowly through childhood and adolescence. Executive dysfunction appears in ADHD, schizophrenia, and frontal lobe damage. Training programs may improve executive functions, though transfer to other tasks remains debated. Cognitive control adjusts processing according to goals and context. Proactive control actively maintains goal information, biasing processing toward relevant features. Reactive control mobilizes after detecting conflict or errors. Individuals vary in their reliance on these control modes, potentially explaining differences in cognitive styles. Mental imagery activates many of the same brain regions as actual perception. Visual imagery engages early visual cortex. Motor imagery activates motor areas. This overlap suggests mental simulation uses perceptual and motor systems in offline mode. Mental practice can improve skills, demonstrating imagery's functional significance. Embodied cognition proposes that thinking involves simulating bodily states and actions. Understanding language about actions activates motor areas. Conceptual knowledge may be grounded in sensory and motor experiences rather than abstract symbols. This framework emphasizes the body's role in shaping thought, challenging traditional views of cognition as purely symbolic computation. Cognitive development progresses through increasingly sophisticated ways of thinking. Piaget proposed stages from sensory motor through formal operations. Current research reveals greater continuity and domain specificity than stage theories suggest. Neural development, including protracted prefrontal maturation, constrains cognitive development. Experience shapes developmental trajectories within biological constraints. Theory of mind, understanding that others have mental states, emerges during early childhood. The temporoparietal junction and medial prefrontal cortex support mentalizing. Autism spectrum disorders involve theory of mind difficulties, though debate continues about specific deficits. Theory of mind allows navigation of social worlds by predicting others' behaviors based on inferred beliefs and desires. Moral cognition involves both emotional and reasoning processes. The ventromedial prefrontal cortex integrates emotional values into decisions. The dorsolateral prefrontal cortex supports abstract moral reasoning. Damage to ventromedial prefrontal cortex impairs moral emotions while preserving moral knowledge, demonstrating dissociable components. Cultural variation in moral values reflects differences in emphasis among universal moral foundations. Risk and reward processing involves complex calculations about uncertain outcomes. The ventral striatum represents reward value and prediction errors. The anterior insula signals risk and potential losses. The prefrontal cortex integrates information to guide decisions. Individual differences in these systems relate to risk-taking propensity and vulnerability to addiction. Metacognition, thinking about thinking, involves monitoring and controlling cognitive processes. The prefrontal cortex supports metacognitive abilities, including judging one's own knowledge and learning. Metacognitive accuracy varies across individuals and improves with age. Metacognition enables effective learning strategies and adaptive behavior. Cognitive biases systematically distort judgment and decision-making. Confirmation bias seeks information supporting existing beliefs. Availability bias overweights easily recalled examples. Anchoring bias over-relies on initial information. These biases reflect heuristics that usually work efficiently but sometimes produce errors. Understanding biases may help counteract them, though debiasing proves challenging. The brain processes language hierarchically from sounds to meaning. Primary auditory cortex extracts acoustic features. Superior temporal cortex identifies phonemes and words. Inferior frontal cortex processes syntax. Anterior temporal cortex represents semantic knowledge. These regions work together to comprehend and produce language rapidly and effortlessly. Reading involves mapping visual word forms to phonological and semantic representations. The visual word form area in left fusiform cortex specializes in recognizing written words. Reading builds on spoken language systems, requiring learning to connect written symbols to sounds and meanings. Dyslexia involves impairments in phonological processing, affecting reading acquisition. Speech production requires coordinating respiration, phonation, and articulation. Motor cortex controls articulatory muscles with exquisite precision. The cerebellum times speech sounds. Broca's area plans motor sequences for speech. Speaking occurs so automatically that conscious awareness lags behind actual articulation. The brain represents space through multiple coordinate frames. Egocentric representations encode locations relative to the body. Allocentric representations encode absolute positions independent of viewpoint. The hippocampus supports allocentric representations, while parietal cortex handles egocentric coding. Transforming between reference frames enables flexible navigation. Object recognition involves extracting invariant features despite changes in viewing conditions. The ventral visual stream processes object identity through increasingly complex feature detectors. Early areas respond to edges and orientations. Later areas represent complete objects invariant to position and size. This hierarchical processing builds complex representations from simple features. Face perception involves configural processing, integrating spatial relationships among features. The fusiform face area responds selectively to faces. The superior temporal sulcus analyzes changeable aspects like expressions and gaze. The amygdala processes emotional significance of faces. This network enables the remarkable human ability to recognize and interpret faces. Motion perception involves specialized brain areas including MT and MST. These regions contain direction-selective neurons responding to moving stimuli. Motion processing supports object recognition, spatial navigation, and predicting future positions. Motion blindness following MT damage demonstrates this region's critical role. The brain fills in missing information using context and expectations. Blind spots where the optic nerve exits contain no photoreceptors, yet you perceive continuous vision because the brain fills these gaps. Similarly, visual completion occurs for partially occluded objects. This filling in demonstrates active construction of perception. Change blindness, failure to notice large changes in visual scenes, reveals attention's importance for conscious perception. Without attending to changing features, even dramatic alterations go unnoticed. This phenomenon shows that perception is far more limited than subjective experience suggests. We perceive a rich visual world, but detailed representations exist only for attended regions. The cocktail party effect demonstrates selective attention's power. You can focus on one conversation in a noisy room, filtering out other voices. Unattended information receives minimal processing, though personally relevant stimuli like your name can capture attention. This selective processing balances focusing on current goals with monitoring for important information. Multitasking often involves rapid switching between tasks rather than true parallel processing. Performance degrades when simultaneously performing demanding tasks. Task switching incurs costs as the brain reconfigures control settings. Some tasks become automatic with practice, reducing interference, but complex tasks generally require sequential processing. Implicit learning acquires knowledge without conscious awareness. Statistical learning extracts patterns from sequences automatically. Procedural learning develops skills outside awareness. Artificial grammar learning occurs without explicit rule knowledge. These learning types demonstrate that not all learning requires consciousness. Priming occurs when exposure to stimuli influences later responses. Perceptual priming facilitates processing of previously encountered stimuli. Semantic priming spreads activation to related concepts. Priming operates unconsciously yet affects behavior, revealing implicit memory's influence. Priming effects can last minutes to hours depending on conditions. Recognition memory involves two components, familiarity and recollection. Familiarity provides a sense of having encountered something before without contextual details. Recollection retrieves specific episodic information. The parahynal cortex supports familiarity while hippocampus enables recollection. These components can be dissociated through brain damage or experimental manipulations. False memories demonstrate memory's reconstructive nature. Misleading questions can implant non-existent details. Imagining events can create false memories of experiencing them. Schemas and expectations influence what is remembered. These phenomena show memory as active reconstruction rather than playback of recorded events. Memory consolidation stabilizes new memories, requiring hours to days. Systems consolidation transfers memories from hippocampus to cortex over extended periods. Sleep facilitates consolidation, particularly slow-wave sleep for declarative memories and REM sleep for procedural skills. Disrupting consolidation impairs memory formation. Retrieval cues trigger memory recall. Contextual cues present during encoding facilitate later retrieval. The encoding specificity principle states that memory is best when retrieval conditions match encoding conditions. State-dependent memory shows that memories are more accessible in the same physiological state as during encoding. Forgetting reflects multiple processes beyond simple decay. Interference occurs when similar memories compete. Retrieval-induced forgetting shows that recalling some information suppresses related memories. Motivated forgetting may occur for unpleasant memories. Forgetting can be adaptive, removing outdated or irrelevant information. Prospective memory, remembering to perform intended actions, involves distinct processes from retrospective memory. Event-based prospective memory triggers when encountering specific cues. Time-based prospective memory requires monitoring time. The prefrontal cortex and hippocampus support prospective memory, with aging particularly affecting this ability. Source memory, remembering where information was learned, depends on hippocampal cortical interactions. Source amnesia involves losing context while retaining facts. False attribution occurs when memories are assigned to incorrect sources. Source monitoring requires distinguishing internally generated information from external experiences. Tip-of-the-tongue states occur when you know something but cannot retrieve it. These frustrating experiences reflect partial activation of memory traces. The target information often comes to mind shortly after searching stops. TOT states reveal distinctions between knowing that you know and accessing specific knowledge. Spatial memory represents locations and routes through environments. Cognitive maps in the hippocampus encode spatial relationships. Landmark knowledge, route knowledge, and survey knowledge represent different levels of spatial understanding. Individual differences in spatial ability relate to hippocampal structure and function. Long-term potentiation provides a cellular mechanism for learning. High-frequency stimulation strengthens synaptic connections persistently. LTP involves NMDA receptor activation, calcium influx, and downstream changes in synaptic strength. This process likely underlies memory formation, though relating cellular mechanisms to behavior remains challenging. Long-term depression synaptic weakening following low-frequency stimulation, balances LTP. LTD removes unnecessary connections and fine-tunes circuits. Both LTP and LTD contribute to learning by adjusting synaptic weights toward optimal values. These opposing processes maintain neural homeostasis while enabling learning. Spike timing-dependent plasticity demonstrates that precise timing of neural activity matters for learning. Synapses strengthen when presynaptic spikes precede postsynaptic spikes. The reverse timing weakens synapses. This rule captures causal relationships between neurons, potentially implementing sophisticated learning algorithms. Neuromodulation adjusts neural circuit function through diffuse release of neurotransmitters. Neuromodulators including dopamine, serotonin, norepinephrine, and acetylcholine affect learning, attention, arousal, and mood. Rather than providing point-to-point communication like glutamate and GABA, neuromodulators broadcast signals that adjust entire circuits. This allows flexible adaptation of neural processing to behavioral state. The brain's energy demands require efficient resource allocation. Only a fraction of neurons activate simultaneously, maintaining energy budget. Active neurons consume dramatically more energy than resting ones. The brain prioritizes energy for neurons performing currently relevant computations, adjusting resource distribution dynamically. Glucose metabolism differs between brain regions and cell types. Neurons prefer glucose but can use ketones during fasting. Astrocytes metabolize glucose to lactate, which neurons can use. This metabolic coupling ensures neurons receive adequate fuel. Impaired glucose metabolism contributes to cognitive decline in diabetes and aging. Oxidative stress results from reactive oxygen species overwhelming antioxidant defenses. The brain's high metabolic rate and lipid content make it vulnerable to oxidative damage. Mitochondrial dysfunction increases with age, elevating oxidative stress. Antioxidants from diet may provide some neuroprotection, though evidence remains mixed. Neurotrophins support neuronal survival, growth, and differentiation. Brain-derived neurotrophic factor enhances synaptic plasticity and neurogenesis. Nerve growth factor supports cholinergic neurons. Neurotrophin 3 affects proprioceptive neurons. Reduced neurotrophin signaling may contribute to neurodegeneration, making these factors potential therapeutic targets. Inflammation in the brain can be both protective and harmful. Acute inflammation helps clear pathogens and debris. Chronic neuroinflammation contributes to neurodegeneration and may underlie depression in some patients. Microglia and astrocytes mediate inflammation, shifting between pro- and anti-inflammatory states depending on signals they receive. The blood-brain barrier maintains brain homeostasis but can be disrupted. Inflammation, trauma, or disease can increase barrier permeability, allowing harmful substances to enter. Some pathogens specifically target barrier transport systems. Understanding barrier function helps design drugs that can reach the brain and protect against pathological barrier breakdown. Brain pH regulation maintains optimal conditions for neural function. Carbonic anhydrase enzymes control pH by managing carbon dioxide and bicarbonate. Acidosis impairs neural activity and can occur during seizures or ischemia. The brain closely regulates pH despite metabolic activity that produces acids. Temperature affects brain function, with neural activity increasing with temperature within physiological range. Hypothermia slows neural processes and can be neuroprotective during surgery or after cardiac arrest. Hyperthermia accelerates metabolism and can damage neurons. The brain includes temperature sensors and thermoregulatory circuits maintaining optimal temperature. The brain exhibits critical dynamics, operating near phase transitions between order and disorder. This criticality may optimize information processing, allowing both stability and flexibility. Neural avalanches, bursts of activity with scale-free size distributions, suggest criticality. Operating near criticality could maximize computational capacity while maintaining stability. Network topology affects information flow through brain circuits. Small world networks, combining local clustering with long-range connections, characterize brain organization. This architecture allows efficient local processing while enabling global integration. Hub regions with extensive connections play disproportionate roles in network function. Oscillations coordinate activity across brain regions. Phase synchrony allows neural populations to communicate effectively by timing firing to coincide with receiver neurons' high excitability phases. Different frequency bands support distinct functions, with theta coordinating longer-range interactions and gamma supporting local processing. Cross-frequency coupling links activity across timescales. The brain's complexity emerges from interactions among simple components. Individual neurons follow simple rules, yet networks exhibit rich dynamics supporting cognition. Emergent properties cannot be understood from components alone, but require analyzing system-level interactions. This emergence characterizes complex systems generally, not just brains. Computational models help understand brain function by formalizing theories and making testable predictions. Biophysical models simulate detailed neuron properties. Mean field models abstract to population dynamics. Cognitive models focus on information processing without neural implementation details. Each modeling approach offers unique insights into brain function. Sleep peacefully, knowing your brain carries out its nightly maintenance, restoring your mental energy and processing experiences into lasting memories. This extraordinary organ that makes thought, feeling, and consciousness possible continues its tireless work throughout the night, ensuring you awaken refreshed and ready for new experiences tomorrow. Your brain represents perhaps the most complex structure in the known universe, containing more connections than there are stars in the Milky Way galaxy. Each moment of experience involves coordinated activity across millions of neurons, creating patterns that encode perception, thought, and emotion. As you drift into deeper sleep, these neural networks continue their essential work, though consciousness fades into dreams and eventually into the profound quiet of deep sleep stages. The mysteries of consciousness remain among the deepest questions facing science. How does the physical activity of neurons give rise to subjective experience? Why does it feel like something to be you? These questions have puzzled philosophers and scientists for centuries. Perhaps future discoveries will illuminate how consciousness emerges from neural complexity, or perhaps consciousness will forever retain elements beyond scientific explanation. For now, consciousness remains a profound mystery housed within the brain. Your brain never truly rests, maintaining essential functions continuously throughout life. Even in the deepest sleep, the brainstem monitors breathing and heart rate, the hypothalamus regulates body temperature, and the hippocampus consolidates memories. This ceaseless activity ensures your survival and well-being, operating automatically without requiring conscious oversight. The distinction between conscious and unconscious brain processes reveals how much happens beneath awareness. Consider how your brain creates your sense of continuous identity despite constant neural changes. Every day, proteins within neurons break down and are replaced. Synaptic connections strengthen and weaken. Neural circuits reorganize based on experience. Yet through all these changes, you remain yourself, maintaining memories, personality, and sense of identity. The brain somehow creates psychological continuity despite underlying physical flux. the future of neuroscience holds extraordinary possibilities. Brain-machine interfaces may restore movement to paralyzed individuals or vision to the blind. Optogenetic therapies might treat neurological and psychiatric disorders with unprecedented precision. Artificial intelligence inspired by neural computation may achieve human-level cognition. Yet with these possibilities come profound ethical questions about enhancement, identity, and what it means to be human. Understanding the brain illuminates human nature and the human condition. Our thoughts, emotions, decisions, and behaviors all emerge from neural processes. Mental illnesses reflect brain dysfunction rather than character flaws. This neuroscientific perspective can reduce stigma while guiding more effective treatments. At the same time, recognizing the brain basis of human nature raises questions about free will, responsibility, and moral judgment that lack easy answers. Every thought you have, every memory you cherish, every sensation you experience exists because of your brain's remarkable activity. This 3-pound universe creates your inner world, your sense of self, your relationships with others. The brain enables everything that makes life meaningful: love, creativity, curiosity, wonder, joy. Understanding how this extraordinary organ works enriches appreciation for the gift of consciousness. As you rest now, let yourself marvel at the complexity housed within your skull. Billions of neurons communicate through trillions of connections, creating the symphony of activity that is your mind. This neural orchestra plays continuously, sometimes producing conscious experience and sometimes operating beneath awareness, but always sustaining the remarkable phenomenon that is you. Rest deeply tonight, trusting in your brain's ability to restore and renew itself through sleep. The facts and knowledge you have absorbed about this remarkable organ now join the vast stores of information held within your neural networks. Your understanding of the brain that understands itself has deepened, creating new connections and strengthening existing ones. Sleep well and awaken tomorrow with renewed appreciation for the extraordinary biological computer that makes your existence possible. Your brain, working tirelessly through the night, ensures you greet the new day refreshed, restored, and ready to think, feel, and experience the world once again. The journey through understanding your brain continues with each waking moment and each sleeping hour, as this remarkable organ learns, grows, and adapts throughout your lifetime. Each discovery about the brain reveals new wonders and new questions, inviting continued exploration into the nature of mind and consciousness. May your dreams tonight reflect the incredible complexity and beauty of the neural processes that create them.